Happy weekend and welcome to the video on problems building cladograms or based on shared characteristics. So Friday in class, we discussed how scientists build cladograms using characteristics that organisms share and characteristics that they do not share. And that helps them build cladograms and to see evolutionary relationships between organisms. Cladograms are really no longer built using shared characteristics. They're built using DNA evidence. When they're built using DNA evidence, they're usually called phylogenetic trees, as I'm in school on the snow day, because I'm more productive at school than I am at home sometimes. So, let's begin talking about why building cladograms based on shared characteristics is not a good idea, and what are the problems with it, and why current cladograms, or phylogenetic trees, as they're called now, are built using DNA data. So first of all, there's bias. You're picking the shared characteristics. And this may lead you to pick traits that are more obvious to the eye than others. Like, you can see things like wings, you can see things like size of eyes, but there are other traits that are not so obvious to the eye that you might not pick. So there's bias in the traits that you pick when you're building cladograms. The next issue with building cladograms with shared characteristics, this one is probably the most important. And it's confusion between structures that are homologous and structures that are analogous, okay? Now, the difference between these two is very, 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 very important, okay? Homologous structures, okay, are traits that are shared by two different organisms from a common ancestor, okay? It means that there's an evolutionary relationship, okay, between these two organisms, and they both sort of got the trait at the same time, okay? Analogous structures, which are due to convergent evolution, which we're going to talk about in a second, right, are structures that look the same, but they didn't evolve at the same time. They didn't get them together. Okay, we're going to look at examples of these to make it make more sense. Homologous structures are structures in organisms that have a similar structure, similar development, often have different functions, okay, and developed or are evidence that the two organisms have a close evolutionary relationship, where they're closely related to each other because they evolved that characteristic from a common ancestor. Okay, the classic example of this, okay, is the arm of a human, the leg of a kitty. Yeah, let's go back and see the kitty here. Hello, kitty. Okay, moving on. The flipper of a whale and the wing of a bat. Okay, if you take a look at the bone structure, okay, of all these structures, they're very similar. Okay, these structures all evolved from a common ancestor. However, okay, they serve a very different function. Okay, the arm of a human, the leg of a cat, the flipper of a whale, and the wing of a bird have different functions. You know, flight, swimming, walking, grabbing things. Okay, their functions are different, but they're structurally the same, okay, and they evolve from a common ancestor. That makes them homologous structures, and it means that these organisms have a close evolutionary relationship. Okay. Now, the opposite of homologous structures are analogous structures. Analogous structures have separate evolutionary histories. These structures were evolved independently of each other. They often have similar functions, similar external form, but they look different internally. But the most important thing is they have a different origin, okay? And there's no evolutionary relationship, okay? So examples of this are wings of insects and wings of birds, okay? They have similar outside structure, okay, but their internal structure is very, very, very different, and they didn't evolve from a common ancestor, okay? There's this idea that these two organisms solved a problem, whatever evolutionary problem they needed, and they needed to develop flight with a similar solution, okay? But they did not evolve that solution together. There is no common ancestor. Now, because there's confusion between these two structures, analogous structures and homologous structures, it's hard sometimes to build cladograms with shared characteristics because you don't know which one of the structures it is. One homologous structures show a close evolutionary relationship, and analogous structures do not. And this can lead to confusion when building cladograms. Okay. Now, analogous structures are thought to evolve from convergent evolution. Okay. And convergent evolution, okay, is when different organisms find a similar solution to a similar problem, but they didn't find it together. Okay? Structures, or analogous structures that evolve through convergent evolution, do not mean that the organisms have a common ancestor. They found the solution of wings, okay, separately from each other, and they do not have a common ancestor. Now, you might have needed to just write that down. I forget there's a blank for that. Okay, so because of these problems, mostly the homologous analogous structure problem, cladograms are no longer built using shared characteristics. Okay, now differences in DNA and protein sequences are used, and when you're building a cladogram using DNA and protein sequence, often it's called a phylogenetic tree. Okay, now this next idea is a little bit complicated. We need to talk about the idea of a molecular clock. Okay, the molecular clock idea or hypothesis, okay, suggests that mutations occur at a calculatable, reliable, or constant rate. Okay? So scientists have calculated, okay, that if you're looking at a portion of the hemoglobin molecule, okay, and you're looking at that protein in different organisms that evolved during different different times of the history of the Earth, they've been able to calculate that the protein hemoglobin undergoes, or the DNA of the protein hemoglobin undergoes mutations at a rate of 0.56 changes per base pair per billion years. Okay? And you can use, and we're not going to do the math behind this, you need to understand the concept. You can use this rate of mutation to calculate how far apart two organisms are from each other. Okay? Like if they have two changes, right? You can calculate that's like 4 billion years apart then. Because 0.56 goes into 2 about 4 times, and it's per billion year, so that would be 4 billion years apart from each other. But you can use the number of mutations that occur between two organisms in a protein, okay, to date how far apart two species split from each other. The more mutations there are, the more distantly related they are. Okay. Now, you may have a question here. Well, mutations, we learned before Christmas, and that seems like so long ago, and so much has changed since then, that mutations can be harmful. And if a mutation occurs, then the codon will be changed, then the amino acid will be changed, and then the protein will fold differently, and then it won't function, okay? But we also learned, okay, that many times mutations have no effect. We learned of lots of times mutations can have no effect. If they occur in the third base of a codon, okay? If they occur in an intron, which doesn't code for a protein. If they occur in junk DNA. If they do change an amino acid, but that amino acid does not affect how the protein folds. The protein still functions the same. Okay? We learned before Christmas, or if the other one, just because I want to bring them all out there, is that if it occurs before the start and stop codon. Or out of the reading frame, not in the reading frame of the protein. All of those mutations have no effect. And we can track those mutations. And since mutations occur at a regular rate, okay, the more mutations and more differences between two organisms in a protein, the more distantly related they are. The more similar the DNA sequences, the closer related they are. So if we take a look at this example here, here we can see the common answer of these 4 organisms, and you can see these 2 here have one difference in the DNA sequence of a protein that they're looking at from the common ancestor. And they've been able to calculate, based on the mutation rate of this protein, that because there's one difference, that these organisms here evolved 25 million years later than this one, than its common ancestor. And if we go further in time, we'll see that there's 2 more organisms. Okay, they have 2 DNA differences from the common ancestor. And because of that, they were able to calculate that these organisms evolved 50 million years later than the common ancestor. Here we have another example. here they're looking at differences between organisms in a protein known as cytochrome C. And if you remember about cytochromes, cytochromes are the proteins in the ETC of the cristae in the mitochondria, and their job is to pass electrons that they get from NADH so that H plus can be pumped into the intermembrane space, just to review a little bit. But if you take a look at, so they're comparing this protein, cytochrome C, in various organisms, and they're looking at the differences in the DNA. How many differences are there? Okay. And from that, the more differences there are, these are the number of differences, right? These are the years, okay? You can see the more differences there are, okay, the further they are from the common ancestor, which would be like here, whatever common ancestor they're picking at, which I actually don't know which common ancestor they're looking at here. But the whole point is that the more differences, okay, the more time that has passed, and the further the organism is from the common ancestor. The other thing to point out is that this graph is linear, okay, which suggests that the rate is constant, right? That's why it's a molecular clock, because it's a linear line. Those points are almost on a linear line, which means the rate of mutation is relatively constant. There. Okay, so I know that's a little bit confusing, but let's look at the entire point, okay? If you're trying to see how closely related two organisms or two species of organisms are, okay, you look at the DNA or protein of a conserved protein, okay? What a protein that's conserved, what that means is that it's not under selection pressure, okay? So it's not evolutionary forces are not acting on it, okay? And that all the organisms that you're looking at have that protein, okay? Cytochrome C is one that's looked at. The ATP synthase is often looked at a lot. Hemoglobin is looked at a lot in mammals. So there are certain proteins that are conserved because they're not under selection pressure, and they're common in all organisms. So you pick a protein, okay, and then you look at the number of either DNA differences, DNA-based differences, that's nucleotides, or the number of different amino acids in the sequence, okay? And this is the whole point down here. The whole point, okay? The more differences there are, the more distantly related the organisms are. Okay, because if there's more mutations, it takes more time for mutations to occur, and that means more time has passed since the organisms evolved from each other. Okay, the less differences, okay, the more closely related the organism is, because mutations take time. So if there's less mutations, less time has passed since those two organisms separated from each other in time, and they're more closely related as a result. So this is an example. Okay, and I don't want to make you count, that's a waste of time. But what I did is that this is the ancestor that we're looking at. Okay, and then these are all the organisms. And what I did is I counted, okay, the number of differences, okay, between these organisms and the ancestral cell. Okay, and from this, you can determine who is the furthest away from the ancestral cell, and that's the cat and the lizard. And who's the closest to the ancestral cell? The amoeba. The amoeba has the least number of differences, and the cat and the lizard have the most. Okay, also most closely related to the ancestral cell after the amoeba is the sponge. Monday in class, and this lesson is difficult, we're going to be learning how to build phylogenetic trees using DNA evidence. And I'm sorry, it's difficult. But that's what we'll be doing on Monday. I know there's a lot of homework. I'm sorry, blame the snow, and I'll see you on Monday.